Topic is a culturing method. What are culturing method? Culturing method involve the techniques of growing microorganisms in a special media in the laboratory. How do we prepare this culture solution? Number one, the culture solution called algae is prepared under sterile conditions. 2. Algae, which is also known as the culture solution, is weighed and poured into a conical flask containing distilled water. 3. The solution is then sterilized at a temperature of about 121 degrees Celsius in an autoclave. 4. The solution is then allowed to cool to a temperature of about 45 degrees Celsius in a sterile petri dish. Instruments required for the preparation of culture solution. Number 1 is microscope. Number 2 is petri dish. 3. Test tube. 4. Test tube holder. 5. Microscope slide. 6. Cover slip. 7. Hand lens. 8. Inoculating needles. Now let me show you the pictures of all those instruments. The first one is microscope slide. Microscope slide is a thin flat pieces of glass used to hold objects for examination under a microscope. The second one is microscope. Microscope is an instrument used to see objects that are too small to be seen by the naked eyes. The third one is petri dish. A petri dish is a shallow transparent lidded dish that biologists use to culture cells such as bacterium, fungi, etc. Inoculating needle. Inoculating needle is a laboratory equipment used to transfer and inoculate living microorganisms. Cover slip. I hope you can see the cover slip. It is square in the shape. It is the small squares of glass that cover the specimen placed on the microscope slide. Its own function is to flatten the specimen for better viewing. We have hand lens. What is the function of this hand lens? It's a magnifying lens that is used to produce a magnified image of an object. Test tube. The test tube is the instrument that is used to hold, mix, and to perform scientific experiments. Test tube holder. The test tube holder is an instrument that is used to hold the test tube. Now, how do we identify microorganisms? Microorganisms can be identified in the air, water, river, and stream by preparing a culture medium. After then, you introduce the sample and incubate it at a room temperature for about 24 to 48 hours. After which it is then observed for growth and characteristics of the colonies. Characteristics of the colonies such as colony pattern, appearance, etc. Ways through which microorganism enters the body. Number 1 is through mouth. Microorganisms can get into our mouth through contaminated food or drink, such as spoiled food and drink. Number 2 is through nose. Microorganisms can enter the body through the nose by breathing in of aerosols. What are aerosols? What are aerosols? Aerosols are in form of insecticides. They are gas that cannot be inhaled anyhow. They are substance enclosed under pressure and released as a fine spray by means of a propellant gas. Like I've said earlier, the example is like insecticide. Then 3, through skin. It can get into our body through open wounds. Then number 4 is through the anus. We all know that our anus is an opening. So microorganism can enter through the anus, then through animal bites. When animals like fly bite us, they can transfer microorganism into our body. The sixth one is blood contact, for example through blood and transfusion. Number seven is birth. We all know that the vagina too is an opening, so microorganism can enter through the vagina. Then the last one is through sexual contact, such as through sexual transmitted diseases. Carriers of microorganisms. What are carriers? Carriers are agents that are capable of transferring microorganisms from one place to another. Non-living things that are carriers of microorganisms are air, water, and food, while the living things are animals and are called vectors. Example of carriers: the microorganism and the disease they cause. The first one here is female Anopheles mosquito. The microorganism for female Anopheles mosquito is Plasmodium, and the disease it causes is malaria fever. The second one here is tsetse fly. The microorganism of tsetse fly is Trypanosome, and the disease it causes is sleeping sickness. The third one is housefly. The microorganism of housefly is Vibrio cholera, and the disease it causes is cholera and typhoid. The fourth one is rats. The microorganism of rat is Rickettsia. Everybody pronounce it Rickettsia. Rickettsia. And the disease it causes is typhus. The fifth one here is Aedes mosquito. The microorganism for Aedes mosquito is virus, and the disease it causes is yellow fever and dengue fever. The sixth one here is rat fleas. The microorganism for rat fleas is a bacterium, and the disease it causes is a plague. I hope with the explanation that I've made, you've been able to you'll be able to identify, to define microorganisms, to define culture method, how to prepare culture solution, the instruments used in preparing culture solution, and the carriers of a microorganism. Now this is our assignment. Number one, you are to define microorganisms and where they can be found. 2. List the groups of microorganisms. 3. Explain the preparation of culture media. Thanks for listening. Bye.